Good evening, everybody. Yesterday we completed, that means previous class we completed till mineral nutrition, that is called plant physiology up to transpiration. Today we discuss about mineral nutrition and enzymes. Mineral nutrition, plants are autotrophic in nature. Plants synthesize their own food material by taking two inorganic components, carbon dioxide and water. In the presence of sunlight and chloroplast, they synthesize chlorophyll. They synthesize sugars. For that purpose, for that process, not only for that process, for many processes, they depend on various mineral ions present in the soil. There are so many mineral ions present in the soil, but very few are regarded as mineral nutrients. A mineral ion is said to be mineral nutrient when it obeys the criteria of essentiality proposed by Arnon Stout. Criteria of essentiality has three basic rules. 1. Every mineral ion must show its existence till the completion of growth and reproduction of a plant. 2. Every mineral ion must involve directly in any process. 3. Every mineral ion should not be replaced by another mineral ion easily. Any mineral ion obeys all these three basic rules, then it is said to be a mineral nutrient. 16 mineral nutrients, 9 macro, 7 micro, their importance. Then absorption of salts, active absorption by utilizing metabolic energy. Then biofertilizers, all this comes under this chapter. Now we start with the questions. Necessity of soil for plant growth was experimentally shown by. Necessity of soil for plant growth. Necessity of soil or mineral ions are experimentally shown by a scientist called Woodward. He conducted an experiment by taking three vessels. In one vessel he took rainwater. In other vessel he took water from Thames River. And in third vessel he took soil extract, that means water squeezed from the soil from Hyde Park. He has grown spearmint plant in this and in this. Plants that not grow well in rainwater, they grow a little bit well in Thames River, but they are growing excellently in soil extract. With this he proved that soil or soil minerals are very much useful in the growth of a plant. That is, necessity of soil for plant growth was experimentally shown by Woodward. Next, Woodward conducted experiments on. Just now I explained, Woodward conducted experiments on spearmint plant. He took three vessels. In one vessel he kept rainwater. In other vessel he kept water from Thames River. In third vessel he kept water from the soil extract of Hyde Park. Spearmint plants that not grow well in rainwater, grow a little bit in Thames water, but very well in soil extract water, proving that soil provides not only water but also many mineral ions for the growth of the plant. So Woodward conducted experiments in spearmint plant. Increase in the dry weight of a plant grown in dilute soil solution was shown by. Increase in the dry weight of a plant. A scientist by name de Saussure proposed that plants gain more dry weight when grown in solutions rather than pure water. This is his analysis. A scientist by name de Saussure proposed that plants definitely gain more dry weight when grown in solutions rather than pure water. That means pure water does not contain necessary mineral ions. So whenever diluted solutions or dilute soil solutions or any solution, if they grow, they definitely grow well. That means plant gain more dry weight when grown in solutions rather than pure water. Increase in dry weight of the plant grown in dilute soil solution was experimentally proved by de Saussure. Aristotle's concept is humus hypothesis. What is humus hypothesis? Aristotle proposed humus hypothesis. It shows that soil is the material with which plant is made up of. Soil is the material with which plant is made up of. So, soil is the material with which plant is made up of, or plants are basically made up of soil only, was proposed by Aristotle. Woodward, just now we explained experiment to prove that soil extract is important for plant growth. De Saussure is this one. So, for this the answer is 3. Essential elements were first discovered by. Essential elements were first discovered not by Aristotle, not by Woodward, not by De Saussure, but by Knops. Knops, Sachs and Knops. Sachs and Knops proposed a concept called as hydroponics. Hydroponics is also called soilless culture. That means method of growing plants without soil or in a medium with all necessary nutrients. If all the necessary nutrients are added to a water, then you will definitely make the plants to grow very well. This concept is called hydroponics, and that was proposed by Knops. So, essential elements were first discovered by Sachs and Knops. Growing plants in organic nutrient solution is called. Growing plants in inorganic nutrient solution is called not nutrioponics or geoponics, or it is answer is hydroponics. The method of growing plants without soil or the method of growing plants in inorganic nutrient solutions is very popularly known as hydroponics. So, answer is 4. Very high potassium ion concentration than the surroundings was observed in the algae. To explain active absorption, Nightella plant was kept in solutions. After some time, Nightella observed large number of potassium ions. After some time, when it is observed, the potassium ion concentration in Nightella plant is several times more than the surrounding solutions. So, this proves that active absorption, wherever metabolic energy is utilized, the absorption of ions is maximum even against concentration gradients. That means, even necessary or not, they are absorbed very much. So, very high potassium ion concentrations than the surroundings was observed in this algae is Nightella. Third option. The chemical nature of substances that carry ions during active absorption of salts. The chemical nature of substances that carry. This is a cell membrane. This is outer space, that is apoplast. It is inner space, that is symplast. Here special carriers are present. Here ions are present. These carriers take the ions and become ion carrier complexes. Once they come here, they show conventional changes and liberate the ions inside the inner space and comes out without any change. These are popularly called as protein carriers. The special carriers which takes ions from outer space to inner space, particularly only in unidirectional manner, that is only from outer space to inner space. Unidirectional manner are proteins which takes the ions from outer space to inner space, By utilizing ATP, they become active. They take ions and become ion carrier complexes, then throw the ions inside the cell by showing conventional changes. So, the chemical nature of substances that carry ions during active absorption are not carbohydrates or sugars or fats, but they are proteins. Uniporters are the carrier proteins that carry. Uniporters are carrier proteins that carry 2 ions of the same time at the same time is wrong. 2 ions are different types at a time is wrong. Several ions of the same type at the same time is wrong. An ion of only one type at a time. Uniporters always take ions either from outside to inside or inside to outside, only one type of ion from only one direction. There is a co-transport. Some carriers take two different ions either in the same direction or in opposite direction. If they take two different ions in the same direction, it is called symport. If they transport ions in opposite direction, that means some ions are coming inside and some ions are thrown out, that is called antiport. So here uniporters are the carrier proteins that carry an ion of only one type at a time. Is the carrier protein that is involved in stomatal movements is the carrier protein that involves in stomatal movements is first protein is always H plus ATPase, which helps in uniporter. So H plus ATPase always helps in uniporter system. By hydrolyzing the ATP, they take ions from inner space to outer space to maintain a proton gradient, so that protons again move back with the help of carriers, showing co-transport with the symport and antiport. So the carrier protein that is involved in stomatal movements is H plus ATPase. This is the carrier porter, not Na plus ATPase or Ca minus ATPase, Mg plus ATPase. It is H plus ATPase. So answer is second option. If a carrier protein carries two ions in the two ions of different types at a same time in the same direction, it is called. So if a carrier takes ions only in one direction, it is called unipole. If a carrier taking some protons inside and along with protons, if it takes Cl minus or NO3 minus, something like that, it is called symport, because it is carrying the ions only in the same direction. If a carrier, some carriers taking H plus inside the cell, when they are taking H plus inside the cell, they throw sodium or some other ions outside the cell. This is called antiport. Both these are combinedly called co-transport, because one carrier performing the movement of two different types of ions either in the same direction, symport, or in the opposite directions, antiport. So here the question is, name the carrier. Here the question is, the carrier, if a carrier protein carries two ions of different types at the same time in the same direction, then it is popularly known as not uniport, or co-transporter is a combination of both symporter and antiporter. So here the answer is symporter, third one. Next, more specifically, if a carrier protein carries one ion from cytosol to apoplast and another one from apoplast to cytosol, it is known as. So some carrier proteins takes ions from like this. So this is, if a carrier protein carries some ions, one ion from cytosol to apoplast, and another from apoplast to cytosol, that means in opposite directions. Apoplast to cytosol is proton, cytosol to apoplast is some sodium, calcium, something like that. So it is the carriers which are carrying two different types of ions in opposite directions. This is popularly called antiporter. A question is asked like this: the salt resistant plants get rid of excess of sodium. So question will be like this, some EAMCET question: salt resistant plants get rid of excess of sodium by H plus, Na plus, antiport. This is the answer. So salt resistant plants get rid of excess of sodium by which of the following methods is the question. So salt resistant plants get rid of excess of sodium by which of the following methods? Means H plus, Na plus, antiport. So more specifically, if a carrier protein carries one ion from cytosol to apoplast and another ion from apoplast to cytosol, it is occurring in opposite directions. That's why it is called antiporter. Next. A carrier protein shows conformational changes only when. Conformational changes means the phase will be towards the outer space when it takes ions. Once it starts moving through the plasma membrane, the phase will change towards the inner space. So that is called a carrier protein showing conformational changes when ATP is hydrolyzed is wrong, ion is released from the carrier is wrong, all of the above is wrong, ion is bounded to the carrier. Whenever ion bounded to the carrier, this will become ion carrier complex. Now ion carrier complex passes through the cell membrane. At one point of time, it shows conformational changes. The phase of the carrier turns from outer space to inner space to release the ions into the inner space. So a carrier protein shows conformational changes only when ion is bound to the carrier. So second option. In H plus ATPase pump, the porters involved in primary active transport. In H plus ATPase pump, the porters involved in primary active transport. Movement of protons from inner space to outer space through a cell membrane is popularly called unipore. So movement of ions by unipore requires special energy called ATP hydrolysis. This method is called primary active transport, and in H plus ATPase pump, the porters involved in primary active transport are not symporters, not antiporters, not co-transporters, but uniporters. So answer is 4. Next. In H plus ATPase pump, ATP is utilized during the involvement of. Every time ATP hydrolysis occurs only when primary active transport occurs. Primary active transport is nothing but uniporter. So in H plus ATPase pump, ATP is utilized during the involvement of first option, uniporters. Next. Antiporters of H plus ATPase pump expel the following ions from cytosol. Antiporters of H plus ATPase pump expel the following ions from cytosols, not PO4. PO4, NO3 minus, phosphates, nitrates, chlorides always involve in symporter. Sodium always involves in antiporter. So antiporter means sodium. That means first option. Antiporters of H plus ATPase pump expel the following ions from cytosol is first option, that is sodium. Next, bacterization means. Now we will come to the biofertilizers. Bacterization means production of bacteria is wrong. Using bacteria as biofertilizer is wrong. Dressing the seed of crop plants with biofertilizer bacteria is correct. Bacterization means dressing the seed of crop plants with biofertilizer. Killing of the harmful bacteria after harvesting is also wrong. Dressing the seed of crop plants with biofertilizers. Bacterization is normally done with the help of or by using 3 bacteria. One is free living bacteria like Azotobacter, symbiotic bacteria like Rhizobium, associated bacteria like Azospirillum. Free living bacteria means living in the soil freely. Symbiotic bacteria means growing inside the roots. Associated means it is whether in the rhizosphere or rhizoplane. That means in the soil around the root or on the surface of the root. Azospirillum increases yield in sorghum, bajra, pearl millet and other things. And azospirillum not only helps in fixing nitrogen but also produce plant growth regulators. So in bacterization, azospirillum plays a very important role. So bacterization refers to dressing the seed of crop plants with biofertilizer bacteria. So 16th question answer is 3. Next. Arrange the following in a sequence of non-essential elements: micronutrient, macronutrient, and macro non-mineral element. So, first non-essential element, cadmium. Cadmium is a heavy metal which is not non-essential, which is a non-essential element. Then micronutrient. Micronutrient is copper. Copper is a micronutrient. Macro mineral element. Macro mineral element is calcium. Macro mineral element is calcium, and non-mineral element is carbon. So, in total 16 mineral nutrients are there. In this 9 are macronutrients, 7 are micronutrients. In this carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, sulfur. These three are called non-mineral essential elements because carbon is hydrogen and oxygen not getting from soil. Carbon from carbon dioxide, hydrogen from water, oxygen directly from the atmosphere, but the remaining 6 are taken directly from the soil. So, non-mineral macro elements are these 3, mineral macro elements are these 6. Micronutrients are iron, zinc, molybdenum, boron, copper, manganese, chlorine. All are mineral micro elements. Micronutrients are also called trace elements. Recently, 2 are discovered as microelements. They are nickel and sodium. So, these are mineral microelements, 9 used in larger amounts, 7 used in minor amounts. So, these are important ones. So, here according to this, cadmium, it is a non-essential element, micronutrient copper, macro mineral element calcium, non-mineral macro element carbon. So, second option. Match the following: list 1, list 2. Essential element, mineral element, micronutrient, non-essential element. Mineral essential element, here answer is 3, A4. Essential element, oxygen. Mineral element is phosphorus, that is B5. Micronutrient, recently discovered micronutrient is nickel, so 2. And non-essential element is bismuth. So, essential element, mineral element, micronutrient, non-essential element, amino acid, nickel, bismuth, oxygen, phosphorus. So, A4, B5, C2, D3 will be the answer for this. So, answer is 3. Next. Match the following again. List 1, list 2. Essential elements, mineral elements, micro elements, macro elements. Essential elements A7. Essential elements are A16, that is A13, not 13, 16, A3. Answer is 4, A3. B1, mineral elements 13, because the remaining 3 are non-mineral elements, so 13. C4, what is C? Micronutrients 7 and macro elements 2. Macro elements are 9. So, essential elements are 16, mineral elements are 13, micro elements are 7, macro elements are 9. So, the answer for this question is A3, B1, C4, D2. So, answer is 4. Now, carrier up by symporter, carried by antiporter, carried by uniporter, carried by co-transporter, fats, nucleic acids, H+, sucrose and sodium. Answer is 2. A4, carried by symporter is sucrose. Carried by antiporter is sodium. Carried by uniporter is protons. And carried by co-transporter is also protons and nucleic acids. Anything protons and sucrose both will come. So, 4, 5, 3 are next. Biofertilizer used for soybean production is R. Biofertilizers used for producing soybean production. Soybean production is improved by, for this answer is third one, not first one, because rhizobium helps in legume nodules and helps in nitrogen fixation, increases production. Glomus also helps in the production of maize, wheat, soybean, potato and red gram. These are the 5 crops by which glomus is increasing. Glomus is nothing but mycorrhiza. So mycorrhizal associations. Biofertilizer used for soybean production is or means 1 and 3. One is rhizobium, which is normally found in leguminous plants. Glomus, particularly a mycorrhiza which helps in increasing phosphate absorption from the soil and thereby increasing production in soybean. So 1 and 3 is the answer. Now, glomus improves phosphorus absorption, protects the host from water stress. Wrong. Azospirillum fixes nitrogen, protects from pathogen. Correct. Azolla enriches the soil with nitrogen. It is a green manure. Rhizobium, nitrogenous nutrition, secrete growth hormones. Rhizobium does not secrete growth hormones, but azospirillum secretes growth hormones. So answer here is 2 and 3. So 2 and 3 is the answer, not 1 and 3, 2 and 3. Azospirillum fixes nitrogen, protection from pathogen. It gives pathogenic nature. Azolla enriches the soil and it is a green manure. Maize, azospirillum, anabina. Azospirillum does not show the association with anabina. Soybean, rhizobium, glomus. Soybean, rhizobium, glomus. Paddy, azolla, nostoc. Paddy, azolla, nostoc. Potato, glomus, azolla. Potato, glomus, azolla. Here answer is maize, azospirillum, anabina, soybean, rhizobium and glomus. So answer is not 3, it is 1. 1 and 2. Protons primary active transport ATP utilization. Protons primary active transport ATP utilization is correct. Sucrose secondary active transport proton motive force. Secondary symport transport proton motive force. Nitrate secondary symport transport ATP. This is wrong. Sodium primary active transport proton motive force. Sodium is not primary active transport. So answer for this is 1 and 2. 1 and 2. Third option is the correct. So 24th is that one. Next. Elements, essential, non-essential elements, micro or macro elements. Iron is essential. It is a micro element is correct. Phosphorus is essential element. Micro element is wrong. It is a macro element. Magnesium non-essential macro element. Non-essential is wrong. It is essential element. Macro element. Manganese essential micro element. So answer is 1 and 4. Fourth option. Iron essential micro element. Phosphorus essential micro is wrong. It is macro. Magnesium non-essential is wrong. Manganese essential micro element. So answer is 4. 1 and 4. Now, essential elements and all. Proton non-mineral macro element. H non-mineral macro element. Correct. Potassium mineral micro element wrong. Boron mineral macro element is wrong. Molybdenum mineral micro element. So answer is second one. 4 and 1 or 1 and 4. H plus is non-mineral macro element. Correct. Potassium mineral element but is not a micro element. It is a macro element. Boron, it is a mineral element but it is not macro element. It is micro element. Molybdenum, it is a mineral element but also a micro element. So answer is 4 and 1. Second option. Now, keep the following biofertilizers in a sequence for the crops of groundnut, potato, wheat, paddy. Groundnut, potato, wheat, paddy. Second one, rhizobium is for groundnut. Third one, glomus is for potato. Fourth one, azospirillum is for wheat and anabina is for paddy. So sequence is for groundnut it is rhizobium. For potato it is a mycorrhiza, that is glomus. Vesicular arbuscular mycorrhiza or arbuscular mycorrhiza. Wheat, the improvement in the crop in the wheat is done by azospirillum. And anabina is always increases the yield in paddy. That is called algalization according to Venkatraman. What is algalization? Growing anabina and nostoc in the rice fields, increasing the yield is called algalization. So For this, the answer is 1. For groundnut, rhizobium. For potato, glomus. For wheat, azoospermillum. And for paddy, anabina. It is a assertion reasoning. Ion carriers exhibit high degree of specificity to the substance or ions. Definitely they show high degree of specificity. What if specificity means? Specificity means ion carriers does not select the ions. They take the ions which are selected by cell. This is called specificity. Ion carriers by themselves don't go to the ions, take them and throw it in the cell. They take the ions only which are selected by cell itself. That is called specificity. Why they are showing specificity means ion carriers are chemically made of proteins. Because they are made up of proteins, they show that specificity. So, ion carriers exhibit high degree of specificity to the substances or ions is correct. Why they are high specific or specificity to ions are ion carriers are chemically made up of proteins. So, first option for this. Next. Assertion. A carrier protein always carries ions from inside to outside. A carrier protein always carries the ions from inside to outside is wrong. They take ions either from outside to inside or inside to outside or only in one direction, that is from inside to outside like uniporters or outside to inside like uniporters. Two ions on the same direction called symporter, two ions in different directions are antiporters. So, a carrier protein always carries the ions from inside to outside is wrong. Uniporters or carrier proteins is correct. So, A is false, R is true for this. Next. All symporters are co-transporters. All symporters are co-transporters. So, co-transporters itself are divided into symporters or antiporters. So, all symporters are definitely co-transporters. Reason, all co-transporters are symporters. You cannot say that all co-transporters are symporters because in co-transporters there are two types, symporters and antiporters. So, all symporters are co-transporters is correct. But all co-transporters are symporters is wrong. So, A is true, R is false. Third option. Carrier proteins undergo conformational changes upon binding of an ion. Carrier proteins undergo conformational changes upon binding of an ion. Once an ion comes and attaches to a carrier protein, the face of the carrier protein will be towards the outer space, that is apoplast. When it is moving inside through the plasma membrane, slowly it changes its phase from outer space to inner space so that it easily throws ions into the cell. So, carrier protein undergo conformational changes upon binding of an ion is correct. Conformational changes exposes the bound ion to the solution on the other side of the membrane. So, because of the conformational changes only, the place in the carrier protein where ion is bounded is definitely sent towards the solution side so that it is easily thrown inside. So, A is correct, R is correct, that is A and R are true, and R is the correct explanation of A. Only by conformational changes the carrier can throw ions inside the cell. So, answer is 1. In H plus ATPase pump, both uniporters and symporters are involved. In H plus ATPase pump, both uniporters and symporters are involved. Transport of H plus ions from symplast to apoplast is known as primary active transport. In H plus ATPase pump, both uniporters and symporters are involved is correct. Transport of H plus from interior symplast to exterior apoplast is known as primary active transport is correct. But the reason is not the correct explanation of A. So, answer will be A and R are correct. R is not the correct explanation of A. That is second option. Now, scientists Daniel Arnon, Sachs, Hoagland, Woodward. Daniel Arnon is known for criteria of essentiality of mineral nutrients. Arnon and Stout proposed criteria of essentiality. What is criteria of essentiality? Every mineral ion, when it follows three basic rules of this concept, it is regarded as mineral nutrients. What are those three basic laws? Every mineral ion must show its existence till the completion of growth and reproduction of a plant. Any mineral ion must directly involve in a metabolic process. A mineral ion should not be replaced by another mineral ion easily. So, Daniel Arnon, known for criteria for the essentiality of mineral elements. Sachs. Sachs and Knopf, known for hydroponics, growing plants without soil or in water with all necessary nutrients. Hoagland. Hoagland is known for absorption of ions. Absorption of ions by plants through root system was first explained by Hoagland. Woodward. Woodward is known for conducting experiments on spearmint plant. Spearmint plant. So, answer is 1. Carboxymethyl cellulose is used for dressing with. Carboxymethyl cellulose is used for rhizobium inoculants are well-known biofertilizers in almost all developing countries. These rhizobium inoculants are added to the seed with the help of a special chemical called carboxymethyl cellulose or gums. When the seeds are added with rhizobium inoculants on their surface with the help of carboxymethyl cellulose, when the seeds are sown in the soil, the seeds germinate and produce new plants which contain unaccountable number of root nodules. So, because of that reason, it is widely practiced in almost all well-developing or developed or growing countries. Carboxymethyl cellulose is a chemical which is used dressing the seeds with not azospirillum or cyanobacteria or mycorrhizal fungi. It is rhizobium inoculants. Instead of showing all the stages, rhizobium coming and attaching to the root, forming infection thread inside the root hair, going to the cortex, secreting phytohormones, making the cortical cells to produce. This is all we study in formation of root nodules in legumes. All those steps are stopped when the seeds are dressed with carboxymethyl cellulose, that is with rhizobium. Those seeds, when starts germinating, they produce directly numerous number of root nodules. Active ion uptake is inhibited in plants in the presence of metabolic inhibitors like cyanides and azides. Why? Because metabolic inhibitors inhibits respiration, which provide ATP necessary for active ion uptake. Whenever cyanides and azides are used and rate of respiration is minimized and ATP formation is stopped, immediately the rate of active absorption also stops. It proves that ATP is very much required for active absorption. So, active ion uptake is inhibited in plants in the presence of metabolic inhibitors is correct. Why? Because metabolic inhibitors inhibit respiration, which provide ATP necessary for active ion uptake is correct. So, both A and R are correct. R is the correct explanation of A, first option. Next, matching. Alkalization, Azolla pinnata, bacterization, rhizobium inoculants. Alkalization means process of use of blue-green algal culture, A2. Azolla pinnata, heavy metal resistant. Azolla pinnata specialty is it can be resistant to cadmium, chromium, so many chemicals in and around factories. So, it is heavy metal resistant. Bacterization, seed dressing with bacteria 1. Rhizobium inoculants, legume crops 4. So, A2 alkalization, process of use of blue-green algal culture. B3 Azolla pinnata is a heavy metal resistant. Bacterization 1, seed dressing with bacteria, and rhizobium inoculants. It is rhizobium inoculants is normally attached to the legume crops to increase the root nodule production. So, first option. Match the following: symbiont, flowering parasite, prokaryotic parasite, chemoautotroph. Sulfur bacteria, rhizobium, xanthomonas, lichen, cuscuta. So, symbiont, rhizobium, it lives in the root nodules of legumes and helps in nitrogen fixation. Flowering parasite is cuscuta. Prokaryotic parasite is lichen. Lichens live. And then chemoautotroph is sulfur bacteria. So, symbiont is rhizobium, Flowering parasite is Cuscuta. Prokaryotic parasite is lichens, and chemoautotroph is sulfur bacteria. Answer is 4 for this. Next. Arrange the following in ascending order with the reference to their number. Maximum possible number of types of essential elements in a plant. Maximum possible number of essential elements in a plant. Number of types of elements called as framework elements. Framework elements only 3: carbon, hydrogen, oxygen. So definitely 2 will come first. Maximum possible number of types of non-essential elements found in plants. Number of types of essential elements derived from the solid matter of the soil. Solid matter of the soil means 13. So answer will be 2. Number of types of elements called framework elements. Next 4. Number of types of essential elements derived from solid matter of the soil. 4. Maximum possible number of types of essential elements will be 13. And 3. Maximum possible number of non-essential elements in the plant. So answer is 2, 4, 1, 3. 2, 4, 1, 3. Next. Driving force of secondary active transport. So driving force for primary active transport, whenever primary active transport takes place, ions move from symplast to apoplast. The driving force for uniporter, that is called uniporter, the driving force for uniporter for the movement of protons from apoplast or from inner space to outer space is popularly called primary active transport, and for primary active transport the driving force or energy is given by ATP hydrolysis. Now so many protons get accumulated in the outer space, proton number in the inner space becomes negligible. This makes a proton gradient. Proton gradient means the number of protons in the inner space is very less, in the outer space is maximum. Now protons wants to come back into the inner space from outer space, but they are not movable to the plasma membrane. They are taken by special carriers. One carrier is taking protons, along with it takes chlorides or nitrites or phosphates or something like that, that is called symport. Sometimes when carrier proton is moving inside, sodium or calcium are thrown outside, it is called antiport. So this is called co-transport. That movement is called antiport. So antiport and symport both are co-transport. This is popularly called secondary active transport. So in primary active transport and secondary active transport, protons H plus are involved. The energy for primary active transport is given by ATP hydrolysis, whereas the energy for secondary active transport is given by proton motive force. When protons are moving very fast, they secrete or they produce lot of energy. So driving force for the secondary active transport is always not energy derived directly from ATP, not concentration gradient, not chemical potential gradient, but it is proton motive force. So these are some of the bits from mineral nutrition which are very much useful. Now we will go to the next chapter, enzymes. So in plant physiology, mineral nutrition, there are so many bits regarding introduction, role of macronutrients, role of micronutrients. For example, potassium and calcium maintain anion and cation balance. Copper is the main component in plastocyanin. Magnesium is present as pectates in middle lamellum. Calcium is present as pectates in middle lamellum. So it's like that, so many calcium, so many mineral ions and their uses, active transport, and then bio fertilizers. Now enzymes. N means within, zymes means yeast cells. The components which are discovered within the yeast cells are called as enzymes. The existence of enzymes was first described in the form of ferments by Louis Pasteur. Later the term enzyme to the ferments in yeast cells given by Kuhne. Later the term zymase enzyme was given by Edward Buchner. Later J.B. Sumner isolated urease enzyme in a pure crystalline form from jack bean plants. He told that all enzymes are proteins, but all proteins are not enzymes. Northrop described about pepsin and trypsin and proved that enzymes are made up of proteins. J.B.S. Haldane proposed a treatise which proves all the enzymes are proteins. Now coming to some of the important bits. Enzymes are proteinaceous biocatalysts. Enzymes are proteinaceous biocatalysts. Enzymes are found in all living organisms and are synthesized in all cell organelles. Enzymes are proteinaceous, biocatalysis 100% correct. But enzymes are found in all living organisms and are synthesized in all cell organelles is wrong. Some cannot synthesize enzymes, some may synthesize enzymes. So, enzymes are found in all living organisms and are synthesized in all cell organelles is wrong. So, A is true, R is false. Third option. An enzyme consisting of only protein part is called. So, enzymes are broadly classified into, based on their proteinaceous nature, divided into two types: simple enzymes, conjugated enzymes. Simple enzyme is with only protein. Conjugated enzyme is with a protein and non-protein part. With a protein and non-protein part. The protein part is called apoenzyme. The non-protein part is called cofactor. The cofactor may be inorganic cofactor like metalloenzymes, organic cofactor. Organic cofactor again two types: loosely arranged coenzyme, very tightly arranged prosthetic group. Very tightly arranged prosthetic group. This is the basic classification of enzymes based on proteinaceous nature. So, apoenzyme cofactor. Inorganic cofactor may be iron for catalase, manganese for IAA oxidase, zinc for carbonic anhydrase, copper for cytochrome C oxidase, something like that. Organic coenzyme NAD, FAD, thymine pyrophosphate, pyridoxyl phosphate. Heme part of peroxidase enzyme is prosthetic group. So, here an enzyme consisting of only protein part is popularly called simple enzyme, first option. Non-protein part of holoenzyme is called, non-protein part, conjugated enzyme is also called holoenzyme because it shows both protein and non-protein part. So, non-protein part of a conjugated enzyme or holoenzyme is called cofactor. It is called cofactor. It is not conjugated enzyme because holoenzyme name itself is conjugated enzyme. Coenzyme is protein, coenzyme is organic part. Here it is non-protein part of holoenzyme is called as cofactor, second option. The cofactor of catalase. The cofactor of catalase enzyme. I will write all the things. Iron is for catalase. Magnesium is for hexokinase. Manganese is for IAA oxidase. Molybdenum is for nitrate reductase. Zinc is for carbonic anhydrase. Then copper is for cytochrome oxidase. That means without these mineral ions, the enzyme cannot show its function. That's why these enzymes are also called metalloenzymes. Iron for catalase, magnesium for hexokinase, manganese for IAA oxidase, molybdenum for nitrate reductase, zinc for carbonic anhydrase, copper for cytochrome C oxidase. So, the cofactor of catalase is iron. Cofactor for catalase is iron.